Hi, this is Christina Howard, and this is a continuation of the Chapter 4 lecture videos, which are about the histological organization of tissues. So I'm going to go ahead and just start where we left off in the last one, which is at connective tissue. And I'm going to do this in the same way that I do my other videos. So it's going to be a combination of me talking and then drawing over the screen here and there. Um, so to facilitate that, I'm going to take my computer apart and throw it into tablet mode real quick. So that might result in you hearing some noises. And then we will get started. Okay, so no, I do not want to switch to tablet mode. Thanks for asking though. Okay, so we just finished epithelium and we talked about the derivatives of epithelium. So this is stuff like glands, which come in a variety of shapes and forms. So if you need a review of glands, you can go back to this lovely figure and just look at the various examples of them. But now we're moving on to connective tissue. So connective tissue is really what it sounds like, which is the kind of glue or structural scaffolding and support that sticks our parts together and also that forms the structural components of our body. So functionally, it is going to bind, support, provide a framework. So let me give you some examples of each of those in their individual capacities. So binds is going to be things like dense irregular connective tissue. Supports also does this. So does things like dense regular CT, areolar CT. Framework, these are things like reticular CT and bone connective tissue. And then protection is stuff like dermis, which is another kind of dense irregular connective tissue. Bone also represents protection because, of course, it's going to protect your soft, squishy organs from anything that might want to damage it. So one important example would be your cranium, which is the set of bones that is fused around your brain to protect your brain from the outside world. So a quick note about the word protection, because I want you guys to be able to understand why that's going to be an acceptable answer sometimes, but not all of the time. Mostly the issue with this word comes from students overusing it in 241 and then carrying that habit with them to 242. So obviously, since I'm teaching 241 right now, I'm not going to have that same carryover issue, but it still makes sense for me to sort of warn you about it. So in lab and sometimes in lecture questions as well, you're often asked to provide a function for a particular tissue, organ, membrane, covering, etc. So what I end up finding is that students really overuse protection and then they just end up using protection as the function for everything, which is not appropriate. So for example, if I give you like, I'm just going to draw some here really quickly, even though it's not a connective tissue. If I draw you a striated muscle tissue so you can see the sarcomeres maybe, etc. And then there's like nucleus sort of crammed over here. So if question one on a lab practical or potentially a conceptual question on a lecture test is something about identifying this skeletal muscle, so ID the tissue, or so this would be a lab question, or lecture question would be something like what tissue is multinucleate and striated? And the answer to both of those things would be skeletal muscle. So in one, you're identifying it visually, and the other, you're conceptually identifying it based on what I describe about it to you. But if the second part of that question, let's say it's 1A, is okay, so you've said skeletal muscle successfully, and then I say provide a function for this tissue, oodles and oodles of students will just write protection, which I abbreviated here to be protect, just because I ran out of room. But like, protection isn't really a function of skeletal muscle. It's certainly not its primary function. So you can't just pick a vague function for everything and universally apply it and expect to get full credit. So you want your functions to be succinct, but also as specific as you can make them for the given tissue that you're discussing. So please just don't make this same mistake. It won't serve you well as you move upward through increasingly complex anatomy and physiology. So this is not me yelling at you, it's me trying to save you from a mistake that might harm your grade later. I want to do everything I can to prevent you from stumbling and give you as much success as I can possibly impart. So we can talk about connective tissue on terms of their actual names. So I mentioned one, so like dense, irregular connective tissue. But sometimes you don't call it this name because there's a specific structure or specific layer that is made out of this thing. So dense irregular connective tissue, the dermis is made of this tissue type plus other stuff in that tissue type, anchored by it, positioned inside of it. So just a quick word on that. You're going to see different ways you can refer to connective tissue depending on what context you're talking about it in. So another aspect of connective tissue is it's usually very vascular. So when you see this word, think arteries and veins. So there's blood vessels in it. There are some connective tissues that are highly innervated. So when you see this word, think nerves. And the dense irregular connective tissue of the dermis is a great example of a highly vascular, very innervated nervous tissue. And if you go to the histology of the integumentary system videos in the lab playlist, I will show you a couple of those innervated structures. Now let's talk about this, relatively acellular. A means without or lacking. So epithelia are highly cellular, meaning that the majority of what you see when you look at the tissue are cells. Comparatively, connective tissues are relatively acellular, so there are cells in them. So it's not the fact that they're completely lacking cells, so it says relatively here. They do have cells, but lots of non-living acellular space between those cells. So what's in that non-living space? Well, matrix, which is just the non-living but organic and biological material that sits between cells. So fibers includes things like collagen, elastic, reticular fibers, and then some kind of ground substance. So examples of ground substance can be proteoglycans. Trying to draw an L in there. There we go. So in cartilage, for example, it can be hydroxyapatite, which is the crystal mineral that makes up most of your bone. It can be just extracellular fluid, which I'm abbreviating as being ECF here. So the way that you understand and diagnose, look at and recognize connective tissue is to note when you look at it, what cell types are there, what kind of matrix there is, so what observable fibers do you see? Do you see a ground substance? Is it obvious? Is it something that's more complicated than just extracellular fluid? And so the combo of cells, the matrix, and then the way all that's arranged is going to be how you identify and distinguish between the various connective tissues. So, connective tissue proper, you can think of it as supportive, so props up variety of organs and other tissues. So you see connective tissue proper. This is one of the connective tissues proper. This is loose areolar connective tissue, for example. It's all over the place. So the top layer of your dermis is made out of this stuff, so it's not dense irregular connective tissue. At the very top, it's actually this stuff, areolar connective tissue. All of your mesenchyme derived body membranes, so your serous membranes, are going to be a thin layer of squamous cells and then some areolar connective tissue underneath of it. And so we have many, many cell types. So I'm going to say diverse. We have a ground substance, which is something. In the case of areolar, the ground substance is, as you can see, clear, and therefore it's extracellular fluid. And then we have lots of different kinds of fibers. So on the left here, we have a little cartoon version, which is showing you sort of what fibers and cell types can exist in this tissue. And then over on the right-hand side, we have an electron, or excuse me, a photomicrograph of what it looks like under an actual light microscope. So what you can see here is that there's a variety of different cell types. Like fibroblasts tend to have pointy lemon-shaped nuclei, whereas macrophages tend to have round nuclei, but that's not universal. You may also see other immune cells like lymphocytes, which are a kind of white blood cell, bopping around in the tissue. What they're there to do is kind of inspect and see if there's any bad guys present, so bacteria, etc. And then you're going to find a lot of different kinds of fibers. So these thick pink ones are collagen, and then these skinny dark purple ones are elastic fibers. And there's all kinds of stuff in here. So what you'll notice in your lab packet is that it says know the 9 cell types of connective tissue proper. That's asking you to be able to list the cell types. It's not asking you to be able to identify all of them, but be able to tell me what should be there. So these are the cell types that I'm referring to there. So if we count them, there are 9. So what you're going to want to do is memorize this list and be able to give a basic function for each of these. So make fibers. These are immune cells. These are stem cells. These are also immune cells, just of a different kind. These are involved in inflammation. So mast cells secrete histamine and heparin, which thin your blood and dilate your blood vessels during an inflammatory response. Adipocytes store triglycerides, which are fat cells. And then we have another kind of immune cell. Are you noticing a theme here? There's lots of immune cells in your connective tissue. Besides your lymphatic system and your bloodstream, the primary location where you find immune cells is in your connective tissue. They're sitting there monitoring your connective tissue to make sure that there's no pathogens inside of you. And then finally, melanocytes, which make pigment. And yes, melanocytes are present in connective tissue as well as in epithelium, as in the epidermis. So they're all over the place. They're not limited to the epidermis by any means. Okay, so the ground substance of the matrix, so remember, we're going to just give ourselves a reminder, this is non-living organic material of some kind. And it's usually organic, but not always. So organic here means contains carbon. So it does not mean grown without pesticides, although, hey, fun fact, organic foods, as they are marketed to you at like Whole Foods or the fancy grocery store, they actually are grown with pesticides. If you're curious about that, ask me later. But one common misconception about quote unquote natural and organic foods is that they're grown entirely without pesticides, and that is straight up a lie. Um, More on that if you ask me, but for these intents and purposes, I am actually meaning organic in the classical scientific sense, which is to say it's a carbonaceous compound, so its basis is primarily carbon, not the other thing. Okay, so the ground substance depends on what connective tissue you're looking at. So fluid would be extracellular fluid or plasma, as in blood. Gel is a proteoglycan and glycosaminoglycan mix. So these fancy chemicals that I'm talking about are protein with sugars hanging off of them. And sugars, as you may know, as well as proteins, are very polar, meaning they have lots of pluses and minuses, so partial or full positive and negative charges. And if you have a large branching, let's say you have a big protein with lots of amino acids, and the chain of amino acids is branching as well. So that's what these pink things are. And then hanging off of those are sugars, which I'm going to make be green here. And I'm not going to draw these exhaustively, but you get the idea. So if we have this big sort of branchy tree-like molecule that has lots of positive and negative full or partial charges hanging off of it, if we take the time to remember that water is shaped like this, and the different areas of the water molecule have different partial charges, that means that these charges are going to be attracted to the charges present on the molecule. So what you end up with is, I'm going to make just a dot. So my dot here equals a water molecule, just because I don't want to write H2O over and over again. But these branchy, very polar molecules tend to attract water molecules. And when water molecules are attracted to an organic compound, we call the little sort of shield wall of water around the compound a hydration shell, because the water sits there due to the charges and it kind of limits access of other molecules to that particular charged thing. But another feature of water and any other liquid is, oops, that's supposed to be a Q, not a G. Liquid is incompressible. So if I have a gallon of water, I can't squish that water and make it fit into a cup of water. There's no way for me to do that. I can do it with gas, but I certainly can't do it with water. So if I have a bunch of water and I know I can't compress it, and then I also contain the water by making it be attracted to this very polar thing, what I create is a ground substance that is very resistant to compression. So if I push on it, it doesn't yield or squish down. It'll push back. So the reason I'm taking the time to explain this is because that is the magic and beauty of a gel, is it attracts water in such a way that it turns water into a compression resistant material. So In Jell-O, which is gelatin, which comes from byproducts of the meat industry, we've just taken a particular kind of water attractant protein-based molecule that comes from cows and pigs and horses primarily. And we've ground it up, and then when you mix it with water at your home to make Jell-O, you've turned the water into a loaf of water, which is relatively incompressible. Like, consider Jell-O liquid right after you make it with actual solidified Jell-O. If I put a penny on top of the Jell-O liquid, it's just going to sink right down to the bottom. If I put a penny on top of the Jell-O, it'll sit on top of the Jell-O because the normal forces, the gravity that gives the penny its mass, is being resisted by the gel matrix of the Jell-O, so the penny can't fall through the Jell-O. So if you make really tough and hardy gels, like the one I just drew for you here, that means you can make cartilage, which is load-bearing, so weight sits on top of it. So knees, your meniscus of your knee is made of this stuff, as well as intervertebral discs. So anything that's going to support weight and help to keep your body structurally sound might be made of a gel. So what I'm trying to do here is give you an appreciation for just how hard the gels of your body are working for you. So another example would be plasma in blood. That's not a gel, that's a liquid, but it does important jobs. The calcium phosphate, which are actually called hydroxyapatite. Salts in bone are crystals that give your bone its rigidity. We've also got viscous, sort of goopy fluid in connective tissue proper. So there's extracellular fluid mixed with some other proteins that make it kind of gloppy, and that gives it its properties. And then, of course, as I just mentioned, proteoglycans in cartilage. So these are just examples of the roles that extracellular matrix can play. So when I say that ground substance of a matrix is non-living, it can be tempting to kind of write that off because we think of the cells of the body as doing the majority of the work of the body, and usually that's true. However, if you make a non-living substance that has particular fancy properties, the fancy properties that you impart to the substance can really do a bunch of the work of the tissue. So that's what I'm trying to impress upon you here, is that it's not just the cells that matter, it's also the non-living components of the tissue. So the matrix that I mentioned, matrices is plural of that, the matrices that I mentioned may or may not contain various types of fibers. So there's three overarching types. Collagen fibers are perhaps the most abundant of all of the fiber types. They're all over the place. In lab, you're going to see them mostly as staining pink because the hematoxylin and eosin stain that makes up most of our slides stains them pink. And then there's going to be examples like tendons, ligaments, etc. So these are going to be resistant to strength or shear resistant, meaning that if I pull on these things, they're not going to snap or break. They're going to resist my pulling. Elastic fibers are made of elastin. So this is the protein that makes up elastic fibers. And this is elasticity or stretchiness. So if I want a tissue that I can pull on and it will go back to the shape that it was before I pulled on it, I can put elastic fibers in there and it's more likely to do that. So to prove this to yourself, I would recommend you do what I'm doing right now, which you can imagine, but I'm not going to show you because it looks stupid. So you see how my voice is changing? That's because I'm taking my index finger and I'm pushing the tip of my nose upward, making me have like a pig nose, and it's also making my upper lip go up as well, which is why I sound stupid right now. So you can do this too if you want. This is an interactive lecture. I invite you to do all the dumb stuff that I'm doing, But I think you'll find that if you do this and look in the mirror and then you let go, your nose goes back to the shape it was before. It doesn't get stuck that way, like your aunt told you when you were making faces at her or something. Um, the elastic fibers in the elastic cartilage in your nose are the reason for that. You can also fold your ear over anteriorly, and your ear will snap back to the way it was before. Again, elastic fibers are what you have to thank for that. So the fact that your tissues are stretchy and resilient, elastic fibers. And then reticulin, which are reticular fibers, these are a framework. So this provides a scaffold or place for cells to hang out. So a lot of your squishy, soft organs that nonetheless are distinct from their environment are actually made of or contain reticular fibers just as a network for cells to hang out in. So examples of this, I'm going to change color here, include your liver, your spleen, and your lymph nodes. So they're not especially hardy compared with other organs because they're mostly made of cells and they're soft for that reason. But those cells need something to stick to hang out together, and so reticular fibers form that network. So it's kind of like analogous to if cells that are similar to each other are going to hang out together, maybe they want to all hang out in the same tree. And the tree has branches made of reticulin. And so that gives our cells something to sit on or hang out on in a three-dimensional way. So it just gives our cells like a place to attach to, a place to be, a place to hang out with each other. It's not a rigid network like you would see in bone or what have you. So the cells are free to let go of the branches and move around if they want to, but they don't have to. And so the reticular fibers made of reticulin, which is the blue I'm drawing here, is just a place for the cells, which are all these little purple guys, to sort of chill in. And you'll see that when you look at a section of the spleen or of the liver. So that's the purposes of the fiber types. And then many of the connective tissues have a mixture of these. And the way that that mixture is arranged determines what kind of connective tissue it is. Okay, so let's go through the types. We've got cartilage. Oops, I didn't mean to do that, sorry. So cartilage is a rigid or load-bearing. I keep doing that, my bad. I'm trying to draw, and I need to switch over to my drawing tool. I'm going to make this purple because that's the color that hyaline cartilage usually is. So load-bearing, compression resistant tissue. So let's look at some terminology here. So cond is the root word that means cartilage. So anytime you see cond something, think of cartilage. So an example of that is a region that you learned recently. Hypochondriac region, so beneath cartilage. So that region is the area that's underneath your costal cartilages. So every time you see this root word, you should think about cartilage. Now, I gave you a Latin and Greek roots glossary in the study supplement section of your Canvas shell. So I really recommend using that as you go along and learn this terminology because it's going to help you unlock vocabulary much more quickly and easily than if you just struggle bus it to try and learn them by rote memorization. All of these little word parts have a specific meaning. So if you work on memorizing the word part meanings, that means when you see a new combo package of them, you, even if you haven't seen the word before, stand a better chance of at least having some idea what it means. I'll give you an example of that at the end of this. So Here we have two roots that mean cartilage, and then we have oblast, which sounds like it's Irish, like it's an Irish last name, oblast, or oocyte. So oblast and oocyte, sometimes they're used interchangeably, but really what this means is oblasts are baby versions of an oocyte. So they're both cells. A blank oblast, and I'm using blank on purpose here because here we're talking about chondrocytes, but there's also things like osteoblasts, which are bone blast cells. Um, blast typically means newly differentiated, sorry, my little thingy is moving here, uh, cell that does a particular job. And the job that it does is related to whatever this blank is. And then the oocyte is just the mature version. So a cell that has just turned from being a mesenchymal stem cell into being a cartilage cell that's going to secrete cartilage matrix, that cell is a chondroblast. It's just formed. It just decided what its specialty was. After a while, that chondroblast is going to surround itself with cartilage matrix until it's completely walled off in a hole called a lacuna. Plural is lacunae. And once it's walled off completely and it lives in this little empty space that's kind of like a cell apartment, then we consider it to be a chondrocyte. It's grown up, it's built itself its own little house, and now it is considered mature, and it is now an oocyte. So that's the reason for that terminology difference. So there are collagen and elastic fibers in the proteoglycan matrix, and so this helps to differentiate between the different kinds of cartilage. So if you don't see any obvious collagen or elastic fibers, if there's neither that are visible, then you're looking at hyaline cartilage. If you see a bunch of elastic fibers in the cartilage, which are very, very dark, then you're looking at elastic cartilage. And if you see a bunch of collagen fibers contrasting with the proteoglycan matrix, then you're looking at fibrocartilage. So that makes it pretty easy. So these are relatively acellular. Remember, there's lots of space between cells. It's not that cells aren't there, it's that the cells are not the predominating feature when you look at the tissue. And then they're also avascular, which means no blood vessels. And that's important because cartilage experiences a lot of pushing and pulling and wear and tear, which would break the blood vessels. So instead of having blood vessels, because remember, if I have a block of cartilage, and here's our little chondrocytes and their lacunae, yada, yada. And then, like, imagine I have an artery going through this. So what I'm going to do is draw the matrix first. So if I have a little cube of cartilage here with my chondrocytes in their lacunae, and they're fine because they're small. And also spheres, domes and spheres are very good at resisting compression. So a spherical space in a solid matrix is going to be pretty good at resisting being squished. Tubes are not so good at resisting being squished as it happens. So if I have a tube that is there to transport blood, and this tube runs through my solid, so let's color that in. Right? And then I'll color the middle of it in too. So this is just the tube being embedded in the matrix. So I'll imagine that it's going through this cube, even though I haven't quite drawn it that way. If that makes any sense at all. So if I then put a bunch of normal forces down on this cube, it's going to resist being squished, but it's probably going to squish a little bit. So I might expect to see its outer boundaries kind of bulge out a little bit and then there'd be a dent where I'm squishing, but it wouldn't completely squish down. But what would happen is that I would be also squishing my blood tube, which would make blood flow through it less well. So we can't have squished blood vessels, right? And so we don't want blood vessels to grow through there. Cartilage relies on diffusion from nearby blood vessels to get its oxygen and nutrients, and that works just fine, actually. So the chondrocytes that live in cartilage secrete an anti, which means against, angio, which means artery, genesis, creation, anti-angiogenesis factor. So if you know that anti means against, which most of us do, that's actually part of common spoken English. Angio means artery and genesis means creation. You might remember that from the Bible. Then anti-angiogenesis together means against artery creation. So it's a chemical, which is what we mean when we say factor, that basically actively prevents the growth of arteries in that particular tissue. So again, we can unlock new and unfamiliar words if we're just wise about how we learn and use our root words. Okay, so anti-angiogenesis factor means don't grow arteries, exclamation point. And then it also lacks innervation, because again, if we're going to be routinely squishing this tissue, and sometimes squishing it quite hard, having nerves in there, it would be painful to squish the nerves all the time, right? And we know this because some kinds of injuries involve injuries that accidentally squish nerves and make it painful for us to exist, right? So that's the general idea there. So I promised you I would give you another example of how you can use this magical sort of skill of root word knowledge to unlock unfamiliar vocabulary. So before we move on from cartilage, I will tell it to you. And it is the following, which is amazing. So let me write a word for you. And it might be a word that some of you are familiar with, but a lot of you probably won't be. So if you're not familiar with it, don't pause and go look it up. We're going to work it out together. If you are familiar with it, just enjoy the exercise, okay? Oops, I want this. I like the thicker dot. There we go. So here's a word that you may or may not have seen. It is pronounced achondroplasia. So if you've never seen this word before, you're like, oh, that's a mouthful. I don't know what that means. And if you stopped there, you'd be like, well, too bad for me that I don't know what that means. I'm going to have either have it explained to me or I simply won't know. Not true if you're a good root word and word part learner. So you might say, okay, well, I don't recognize this word, but I do recognize parts of it. So I can say A means lacking or without. Chondro or chond means cartilage. And you might or might not know, but you will learn that plasia or plasis is a root or word part in medical terminology that we use to describe cell growth and proliferation. So if you put all that together, achondroplasia means lacking cartilage, cell growth, and proliferation. So you might be saying to yourself, oh God, it means somebody doesn't have any cartilage. So they're just like bones and squishy bits. That's horrifying. Not quite. During bone growth, when you're going from being a small person to being a large person, the template or the precursor material that you have in place of bone before you turn it into bone is cartilage. So in a typical bone, there's the central area, which is the shaft, and that's usually ossified. And then there's this area of cartilage on either side. And then there's a bony end that also has some cartilage at the joint So this and this is cartilage. So what happens at these little areas, which are called metaphyses, by the way, you'll learn about those in the skeletal system lecture, is as a person grows, the edge of the cartilage that's facing the bone gets turned into more bone. And so we lengthen the bone a little bit that way. And then more cartilage gets added. And then that cartilage gets replaced by bone. And so that's how bones get longer. And therefore how humans get taller. So achondroplasia affects these little cartilage regions where they just don't grow, resulting in permanently short bones. So achondroplasia doesn't mean lacking cartilage entirely. It just means that the parts of the cartilage that are responsible for skeletal system growth don't do what they're supposed to do. And that is a form of genetic dwarfism. And it's the dwarfism style that describes a failure of the cartilage growth plates to actually do their jobs. So, if you had never seen that word before, you might have been thinking to yourself, oh no, I'm just never going to know the answer to it, or I'll have to look it up. But if you're wise about how you use your root words, you can kind of suss it out. And you can also use context clues. So if I was going to ask you a lecture test question about this, for example, I'll give you an example of that as well, because having lecture test question examples is always really nice. So if you'll just bear with me while I write this, and actually, you know what, I'm going to pause the recording while I write it so that you don't have to sit there and watch me. Okay, here, I'm back. Here's my question. So let me read it to you since I kind of wrote it quickly and therefore my handwriting's not very good. A person with achondroplasia remains very short in stature because their blank fails to grow and be replaced with bone. And then we have some choices here. So what you can do, like let's say you're a student and you're taking a test and this is one of the questions and you just don't remember seeing this word. And so you might identify it as kind of the key word here and be like, I don't know. But then you would look at the other parts of the question and say, okay, they're very short, which means they're not growing. So maybe it's something to do with their skeletal system. And then there's a blank. And then I also see that it's replaced with bone. So choice number one is bone. So if I look at that and be like, oh, bone, 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 because it's skeletal system, and then I say like, their bone fails to be replaced with bone, that sentence does not make sense. So it's definitely not that. And then you would go through and say like, to my knowledge, is blood ever replaced with bone? And then you would think about your blood gradually turning into bone inside of your blood vessels and probably become horrified by that prospect. And you'd be like, well, it's not that. And so then you have choice between cartilage and muscle. And if you know that the primary function of muscle tissue is to move, and you know that the root word for muscle is myo, and you then notice, oh wait, I see the word cond here, and also the word A. So even if you're not sure what plasia is, A is what lacking or without, and cond is cartilage. So this word is telling you it's without cartilage, and then there's a blank, and I've already ruled some stuff out. So even if I, a student, didn't know this word before, if I was careful about the question and its context and the root words, I could conceivably, even never having read this word before, still arrive at the correct answer, which is B. So that's an example of how you can use a couple different tools in your testing toolbox to unlock the special superpower of increasing your chances of getting questions right, even if they contain words that you've never seen before. And that's something that's really important for you to be able to do, because for the majority of students who haven't learned this skill, if they encounter a word they don't recognize, that causes their test taking experience to grind to a screeching halt. And it can send them into an anxiety spiral that prevents them from being successful on other questions. So if you know that you have a set of tools in your mental toolbox that you can unlock to avoid those problems, that gives you a huge advantage over other test takers and really can give you an edge up both in class and in your programs, because you're going to take multiple choice tests in 241 and 242, and the multiple choice type of questions as you move through A&P and up into higher division science and eventually into your program culminating, for example, in the NCLEX nursing test, which is a notoriously nasty, hard to pass multiple choice test. If your skill set and your toolbox is full of stuff like what I just showed you, you will be able to pass those tests at higher rates than your peers, than your program cohorts, than your classmates. That's what's going to separate you from the rest of the pack. So that's what I want to help you learn starting right now, how to separate yourself from the pack. Because I know that I want you to leave here with the best chance of becoming rock star, excellent clinicians and not just mediocre ones. And I know that's what you want too, because you're here because you want to do a specific job. And I'm sure that you want to shine in your job. No one's like, I would like to be the world's most mediocre physiotherapist, or I'd like to be a very average nurse. That's my goal. None of you want that, right? So I'm here to help you not only become what you want to be, but become the coolest, most badass version of what you want to be. That's the goal. Okay, so now I'm getting off my little soapbox here and we're going to finish this. So a lot of the tissues that we're going to talk about have a wrapping of some kind. So this root word means around or surrounding. So peri means around or surrounding, and chond means cartilage. So perichondrium literally is just the tissue layer that surrounds cartilage. Voila. So it consists of two layers. There's the outer layer, which is dense irregular connective tissue. And then there's this transitional layer. So this is an area where fibroblasts, which make collagen, experience metaplasia and turn into chondroblasts. And then once they wall themselves off, they move to the center of the tissue and become chondrocytes. And an oocyte is the mature version. So the way that you add to and maintain this tissue is that these cells, so let me make my little pointer smaller here, like this guy, for example, is going to move down and become this guy, his neighbor. And then as the tissue or cartilage grows and more layers are added on top, that cell moves into the center. And then another thing you'll see are these chondrocytes in their lacunae are clustered into little stacks. And they're usually even numbers-ish. So like this one has 4. This one has 2. This one has 2. These are called isogynous, that's a U, cell clusters. Iso means same and genus means creation point. So isogynous cell clusters means all of those cells used to be the same cell, and that cell underwent mitosis a few times to create more of itself. So here we can see 2 that have recently divided, and they've built a little wall between them to give themselves separate lacunae. So this is how cartilage grows and how cartilage is maintained. Okay, so when something is added to at the edge, like I just described, we call it appositional, which is a word that means add material to the edges. So this is the same way, like let's say you're a potter and you're making a pot on a wheel and you realize the wall of your pot is too thin. And you're like, oh man, this is gonna be a really fragile pot if I leave it this way. So what you might do is grab some more clay and add it to the walls to make the pot walls thicker. That would be appositional growth. So here you can see exactly the process that I just described. We have the outer layer of the perichondrium here, and it contains fibroblasts. And then as those fibroblasts move in, they differentiate into chondrocytes, well, first chondroblasts, and then they turn into a full chondrocyte when they're fully ensconced. So they're not still building this unsolidified matrix around them, they're fully surrounded by mature matrix. So you can think of chondroblast, and here I'll write it in a different color, as another way to say young chondrocyte. Those are sort of interchangeable. So That's appositional growth of cartilage. So in the previous example of achondroplasia, in which I said the dwarfism style where the cartilage matrix doesn't grow and therefore it can't be replaced by bone, it's this growth pattern that is being impeded. So interstitial growth is the other component. So remember I showed you those isogenous cell clusters. What they're going to do is one cell, let's say this cell is called Sally. Sally's like, oh boy, time to divide. I think I want a neighbor. And so Sally's going to divide. Here we can see anaphase, for example. And then once Sally has divided into, let's say they switch sexes every generation. So this is Sven and this is Steve, the daughter cells. They're going to start building a wall between them and adding new matrix around them, which eventually pushes them further apart. So let's pretend like Sven and Steve are brothers, but they don't like each other very much. So they're going to add wall between them so that they can move further apart. If all of the isogenous cell clusters in some cartilage are doing this and adding matrix between each other, but also between adjacent cell clusters, that expands the tissue in general. So both appositional and interstitial growth are important. So I described these types earlier, hyaline cartilage, fibrocartilage, and elastic cartilage. Let's just have a look at them. Remember what I said was, these are cartilages that you can divide into having just the proteoglycan matrix or adding some different fibers to the mix to give the cartilage some different properties. So hyaline cartilage is by far the most abundant. Hyaline means glass-like, which as you can see, it's a very pretty tissue. It looks like stained glass. And then it's also the most abundant. So it's partially in your nose, although there is some elastic cartilage in your nose as well to give it that springiness. It's also on the ends of the majority of your bones so that your bones don't grind against each other in your joints. It makes your trachea and bronchi stay open like a drinking straw instead of collapsing. So it just provides strength, flexibility, ease of movement, load bearingness, protection of certain organs like the hypochondriac region. It does all kinds of things because it's so abundant and it's present in so many different locations. And it's very, very beautiful. It's not my most favorite histological tissue, but it's pretty close. I think elastic cartilage is my favorite one. So fibrocartilage is specifically compression and shock resistant extra much, and that's because the strength of collagen fibers, which in this case have been colored blue, has been added to the proteoglycan matrix. So here we have our chondrocyte, and then you can see the outline of the lacuna, but you can also see lots and lots of collagen fibers. Now you might be wondering, why are they blue? All of this stuff is transparent. The color that it turns depends on what stain you choose to use. So instead of staining this particular clump of fibrocartilage with hematoxylin and eosin, maybe it's stained with the azan stain, which stains things blue, or just with methylene blue, which is a blue dye. But regardless, the dye is blue, so stuff will show up blue. So this is just sort of a connection piece where I connect something you're seeing in lecture to something in lab, which is just to say, although most of the histology pictures that you're going to see are going to be pink and purple, that is entirely dependent on the stain that the histologist chose to use, and you're not always going to see pink and purple. Sometimes it looks different. So that's what you're seeing here. So these are basically resistant to compression, so being pushed or squished, and shock. So think about jumping up and then coming back down. When you jump up, you are propelling yourself into the air using your muscles and the energy stored in your Achilles tendon. You hang weightless in the air for a moment, but then gravity, because the earth is turning, is like, nope, you have to go back down because I say so. What goes up must yada, yada, yada. So when you fall back down, you're going to fall with an intensity that's proportional to your mass. And I think you'll find, if you think about it, that the majority of your mass, your heaviness, is above your knees. And that you only have two bones in your lower leg, and then those sit on top of your foot, right? So when you come back down to earth from jumping, especially if you jump high, there's a lot of weight, added gravity as well, coming down on the tops of your tibias, which are the major support bones of your lower legs. Fortunately, your meniscus of your knee is a cup of fibrocartilage that sits at the top of your tibia, and it contains the end of your femur and also cushions that impact. If you didn't have that, jumping would be painful for you. And having a torn meniscus means that you tore your fibrocartilage cup and now it's mad at you, which can cause things like knee dislocations. If you want to be an osteopath or a physical therapist, etc., that might be something that you end up dealing with, is rehabbing people who have ruined their knees. Elastic cartilage is probably my favorite tissue to look at histologically because it's beautiful. So you've got a lot of it in certain places. So the external ear, this is why you can taco your ear and it goes right back to the way it was. I would add to this that there's a small amount in your nose. But regardless, you can see these dark areas here. These are clumps of elastic fibers that give that elastic property to the cartilage. Okay, so now we're going to move on to tissue membranes. So what do I mean by tissue membrane? I mean not a cell membrane. So remember, a cell membrane is a phospholipid bilayer. So that's what you would see like right here, for example. That's not what we're talking about. Tissue membranes are multicellular. And they're basically large sheets that wrap or cover or line something. So what they consist of is some kind of epithelium and then a connective tissue that's underneath. So they can line body cavities, for example, peritoneal cavity, or openings like oral opening. They also cover surfaces, so things like your gastrointestinal tract, but also your integument, which we get our own video about next chapter. So they're there to do a bunch of different things: support, protect, lubricate, all kinds of stuff. So we'll go through each kind. But one thing you're going to need to remember that you learned in Biology 160, but maybe haven't thought about for a while, are cell junctions. So cell junctions are going to do things like allow material to pass between cells. So we can see these are gap junctions where there's a little tube. So if this like chloride ion wants to go through into this cell, it can. We can also see desmosomes. These are sort of like rivets. They hold cells together. And then up here, the cell membranes are quilted together. So these are going to be, oops, I don't want that. There we go. Tight junctions, which basically prevent things from passing between cells. So all of these kinds of cell junctions are going to give the membrane of the tissues the structural integrity that it has. So the additive property of tissue membranes comes a lot from cell membrane interactions as well. So we've got mucous membranes, serous membranes, the cutaneous membrane, and last but not least, the synovial membrane. So these two are most common. And then these two are more specialized. So this one is specialized in the skin or integumentary system, and the synovial membrane is specific to synovial joints, which is part of the skeletal system. So we have two kind of general ones and two specific ones. So one thing you're going to notice, and this is a spelling point, but I want to make you aware of it, is OUS is present in all of these, except for the synovial membrane, you'll notice. So this is a suffix in English that means it's an adjective. So the reason I'm making this very clear is because sometimes you're going to need to refer to something as in its adjectival form, like mucus, but there's also a noun form. So for mucous membrane, the noun is mucus, no O, which is a substance. Specifically, the substance produced by a mucose membrane. Similarly, serous refers to serum, which is a thin watery fluid. Serous means whatever this thing is, membrane in this case, creates serum. So the reason I mention this specifically as it relates to mucus is because sometimes students struggle. They're like, well, should I use the U form or the OU form? I'm not sure which to use. And the answer is it depends on if you're talking about mucus, the stuff you hork up when you're sick, for example, or whether you're talking about it in the adjectival form and you're describing a mucous membrane. That's what I mean. So let's go through the membranes and we'll just talk about kind of where they're found and what they do and all that good stuff. Oops, sorry, didn't mean to do that. So mucous membranes are what they sound like. So I'll give you some examples of areas you can find these. Airway, orifices, GI tract. Just a few examples, also repro tract. So you have some kind of epithelium with mucus secretion. Often that epithelium is columnar and the mucus cells are goblet cells, although not always. But regardless, the commonality is mucus secretion. Serous membranes line things like your pleura, which line your lungs, your peritoneum, which lines your abdominal cavity and your pelvic cavity, and your pericardium. So your lungs, your intestines, your stomach, and your heart are all organs that are made of muscle and they're in motion. And when two things next to each other are in motion, they tend to rub against each other, right? So what you don't want is for things that are going to rub against each other to create friction because friction damages tissues. So if instead you have this transudate, which is a serous fluid, you can create a lubricating layer that allows the organs that are in motion to kind of slide past each other without creating friction, and the tightly joined cells combined with the areolar tissue, which remember is home to many immune cells. If you combine those two, basically you can get a fully lined body compartment that contains organs in motion and keeps that compartment separate from other compartments, but also allows lubrication for the contents of that compartment. So these two I'm actually going to not talk about here because this is chapter 5 and this is chapter 9, which we'll do independently. So you can you can read about these in your textbook in the reading schedule and give yourself an overview, but we're not going to delve into these deeply until another lecture, so I'm going to skip them for now. Okay, so we don't have much more to go. We're about to finish this. I know it's been long. So similarly to the other chapter, the stuff that I just showed you, chapters 5 and 9, for example, for skin and joints respectively, muscle and nervous tissue are what are considered to be electrically excitable. meaning capable of carrying an electrical charge. And so because they have this special property with specific physiology relating to electrical charges, they each get their own chapters as well. So for lab, you have to be able to identify them and then list basic functions. But I want to stress, and I'm going to try and write in italics, even though I realize now that I've started doing that it's not as easy as it seems. Basic functions, not complex. So for muscle, you could put contraction or movement, etc. For nervous tissue, you could say send electrical impulses. So you don't need to know detailed functions until we actually get to those chapters. And you'll find that there's a lot of chapters devoted to these because they take a lot of effort in physiology and chemistry to explain. So more on those later. Okay, so tissue injuries and healing. So we're going to talk about the process of inflammation and then regeneration after that. And then I'm going to give you a little tutorial about the various kinds of plasias because those are important to know as well. So this is kind of a fundamental, oh no, I didn't mean to do that, but actually this is fine. So a fundamental process is we don't exist in a capacity where we can like avoid getting hurt. The world is full of sharp edges and gravel and potential for our skin and our tissues to become injured. So we need a predictable and repeatable set of steps to go through when we have that happen to us that reliably fixes the problem. So that's what we're dealing with here. So the first process is inflammation. And I want to stress, because I know that there's some fads around wellness and healthcare that characterize or blame everything on quote unquote inflammation. And so like if you follow diet or wellness Instagrams, for example, some of those people are absolute kooks and you shouldn't pay attention to them. And they'll just say things like, this smoothie detoxes you and reduces inflammation, and then they don't bother to specify how or why or what properties or even what they're talking about. And if you ask those people to define inflammation, they'll often just describe like a general sense of unwellness or unease. They're not able to give you any specifics, which is a big signal that they have no idea what they're talking about. So regard those people with suspicion, and if appropriate, tell them what I'm about to tell you. So inflammation is not quote unquote bad, although it does hurt. It is necessary and a normal response to tissue injury. And then regeneration is replace or repair. There's an I in there. What was damaged or lost. So sometimes you just straight up lose some tissue. Sometimes it's just messed up in some way, but regardless, you have to grow more cells to replace it. So we'll talk about that as well. Okay. So I'm not going to read this to you. I'm just going to kind of describe all of these aspects. I'll let you read the figure because you're more than capable of reading this to yourself. So obviously this kid has a boo-boo, one that we've probably all got at some point. Having a skinned knee is a pretty normal part of childhood. The problem, though, is that when you break your skin and you remove a piece of the epidermis and you expose the dermis, which contains blood vessels, bacteria covers almost everything we interact with. And so that's a way for pathogens and toxins like bacteria to hitch a ride into places in our body they shouldn't be. And that can create the possibility of us getting an infection, right? So we don't want that. So pathogens, toxins, so things like urushiol. I'm just giving you an example here. Urushiol is the toxin that makes poison ivy and poison oak extremely itchy and blistery waste products and chemicals from injured cells, these are going to stimulate inflammation. So the second question is, how does your body know when there's pathogens or toxins in it? Like, how does it detect when one of those crosses the cutaneous membrane and ends up inside of you? And the answer is mast cells know. So a mast cell is a type of modified immune cell, and all of these little granules you're seeing in there are these: histamine, heparin, and prostaglandins. So these chemicals create the cardinal signs of inflammation. So cardinal means if you see an injury or an area on a patient that meets all these criteria, then that's inflammation, and you can suspect that an inflammatory response to something is going on. So swelling, so when more fluid goes into the tissue and makes it puff up; redness as a result of more blood going to the area; warmth, also more blood; and then pain, which is, you can thank prostaglandins for that. Redness, swelling, heat, and pain are the cardinal signs of inflammation, and yes, they're uncomfortable, but they are good. So here's what happens when a mast cell releases these granules in response to detection. And in order to detect those things, by the way, not pictured here, but they're basically covered in receptors, and that if one of these toxins or bacteria binds to the receptor, it basically sets off alarm bells for the mast cell, which then begins to blorp out these inflammatory chemicals. So one is increased blood flow. So histamine dilates your blood vessels, so your little capillaries and whatnot, and heparin thins your blood. So if you think of blood vessels as a road that cells travel on, if you make the road wider and you make the road less resistant to flowing, it makes it easier for immune cells to essentially arrive at the place where the bad guys might be. And histamine also increases capillary permeability, meaning that stuff has an easier time passing into and out of blood vessels. So blood flow increase is good. This causes redness and swelling, though, and the plasma leakage out of the blood vessels due to the permeability increase causes the swelling. And then prostaglandins primarily are what account for the pain, but pain is an important signal. You need to know if you have a damaged area of tissue because you need to know how to keep that damaged area away from further injury or pathogens, and also keep it safe and clean so that you can heal from it. If you didn't have pain, you wouldn't be able to do those things. So I'll give you some examples of what happens when you don't have that ability after the end of this. But as a result of the mast cell chemicals that I just described, you get this increase in temperature. So that's going to become warm and red, the tissue that's injured is. But also if you need to do mitosis, which is stimulate the reproduction of immune cells and the beginning of reconstruction of cells that are being replaced, and then also the phagocytosis that all of the immune cells do, where they're going to eat up dead tissue and pathogens, that takes a lot of ATP. So you need lots of oxygen and nutrients for that. You've got all kinds of phagocytic activity, including macro and microphages. You might even get some neutrophils from the bloodstream coming in, because neutrophils are really good at eating bacteria specifically. So that's going to clean up any pathogens or debris from the tissue. And then you also want to carry the toxins and wastes away from the site of injury so that they can get dealt with. Specifically, you're going to end up peeing and pooping them out. So the kidneys and the liver are going to turn those toxins and wastes into less harmful products. They're both capable of doing some pretty magic chemistry, and that helps you clear that stuff out of your body. So as that process gets underway, you're going to have an initial period of discomfort, and then after that, the inflammation subsides and give rise to regeneration, which is when the tissue begins to fix itself. That's when we replace what was lost and stabilize things that are healing. And that's going to inhibit mast cell activation. So ultimately, all of this is a negative feedback loop. So the initial response was turn mast cells on, and now we're just going to turn them off again. I am trying to go to the next slide or to escape from this zoomed version. Just 1 second. I'm going to try and fix this because PowerPoint's misbehaving. Okay. So we talked about this already, so I'm just going to skip this slide. I just decided to go through it by zooming in because I had the opportunity to. So this is really just a recap of this, but it does include some kinds of cell death. So you might remember lysosomes. These are going to be, I'm going to stop using red. I want to use some calmer color. These are the recycling center of the cell. Necrosis happens when cells die off schedule, meaning they're not dying via a natural process because they're supposed to. They're being killed by something, either because they were squished or poked or stabbed or a bacterium killed them. But regardless, necrosis results in a cell going from a happy cell with good membrane integrity to broken membrane, organelles all over the place. The lysosomes might burst and release lysosomal enzymes into the tissue, which basically begin to liquefy your tissue because lysosomal enzymes are there to depolymerize all of the things that make up our cells. So we want to avoid necrosis because it's not the correct kind of cell death. So pus may result from this, and this contains cell debris, which is what I just drew here. So random broken cell bits, fluid, dead cells, either entire or more usually this kind, and bacteria. Sometimes they're dead tissue cells, sometimes they're dead immune cells. So pus is actually a combination of your dead little soldiers on the battlefront against pathogens, bacteria, which are trying to eat up the nutrient-rich components of all these dead cells, cell debris, fluid, etc. So even though pus is icky, it's also evidence that necrosis has set in and that you need to clean that area out to prevent any further infection from occurring. So sometimes you get pus in an enclosed space because that process is initiated deep in your tissue, and that's when you get an abscess. And those are really painful for a variety of reasons, but one of them is because they grow and place pressure on surrounding tissues. And they also tend to be really stinky when they pop. So if you work in a emergency department or the dermatologist office, that might be something that you end up dealing with. So we talked about mast cells already. They are basically a specialized immune cell that sits in your connective tissue specifically and releases some chemicals. So histamine does the following. It basically increases diameter of blood vessels, and it increases capillary permeability, which means that plasma exits your blood vessels, but it also means that white blood cells can squeeze out of your blood and into your tissues to go fix the problem. And then heparin thins the blood and prevents clotting. So regeneration starts with fibroblasts. So what they're going to do is lay down collagen fibers, which I'm going to add here. So when we say scar tissue, what we really mean is collagen. So here's the deal. Scar tissue is not supposed to be a permanent solution because it can't replace the function of another tissue type. So, sorry, I was listening to an announcement. I'm recording this from the library since my internet's not very good right now. So the scar tissue basically is just collagen fibers. So it's there to take place of and fill a void and it's there to stabilize the area as it heals. But like a collagen fiber can't replace the function of a melanocyte, for example. So it's not going to be able to make pigment, it's just a fiber. So what that means is scar tissue is a good stabilizer, but it's usually supposed to be replaced eventually with the cells that were there before. However, different organs have different abilities to repair themselves, and that depends on which tissue types are present. So organs that are derived from epithelium, for example, those are two good examples, are highly regenerative. So this is one of the glories of your lungs and your liver. If I smoke and I stop smoking, it's not that I've done damage to my lungs that will never go away. Because over time, as long as I stay quit smoking, my epithelial cells in my lungs are going to turn over and regenerate and replace what was damaged, and my lungs will return to a healthier state. Same deal with my liver. If I donate my liver, I'm not donating my whole liver. I'm giving someone with no liver function, I'm giving them a hunk of my liver, and eventually my liver will grow back to a large extent. And the chunk of liver I donate to the liverless person is probably, assuming it's managed well, also going to grow and get bigger. So epithelially derived organs are really, really good at this. Other kinds, not so much. So fibrosis is one challenge for wound and healing management. Sometimes the signal to lay down collagen doesn't go away, or it isn't interpreted very well by the fibroblasts, and that can create fibrosis. So an extreme example of that is oops, I can't spell and talk at the same time apparently, keloid scar. So if you don't know what those are, look them up. They're common in piercings, for example. So if you see a large bulbous pink deposition of collagen fiber at the site of a piercing, that's a keloid. Basically, at the site of that piercing, the fibroblasts that are trying to heal the piercing injury just never turn off. And so they're going to continue to lay down collagen even long after the hole is healed. So that is a cell signaling problem primarily. So this is the end of the slideshow. I did want to take a moment though, because it's on your study guide and it's an important idea to classify some types of cell growth and cell death. So I mentioned the word plasia, meaning the creation or genesis or regeneration of. So hyperplasia equals tissue growth by an increase in cell number. So cells do this via mitosis. This is not the same as hypertrophy, which is tissue growth by increase in cell size. So let's give some examples of each of these just because it's always nice to be able to sort of understand things from the perspective of their examples. So this would be like literally growing, fetal development, getting larger, etc. And this would be, an example would be building muscle by working out. Metaplasia is conversion of one cell type to another. So sometimes there's normal. So an example of this would be, i.e., a fibroblast turning into a chondroblast. Some metaplasias are pathologic, meaning they're evidence of illness. So an example of a pathologic metaplasia would be your esophagus is supposed to be lined with stratified squamous epithelium, because stratified squamous epithelium is abrasion resistant. If you have prolonged GERD, which is gastroesophageal reflux disease, or an extreme case of bulimia nervosa, you can get the conversion of the esophageal epithelium from squamous to columnar or cuboidal, which doesn't have two L's, just got carried away. So this metaplasia of the esophagus is called Barrett's esophagus, and it's often a warning sign or a precursor of esophageal cancer. So not all metaplasias are pathologic, but some of them are. There's sometimes where a cell is supposed to stay the cell that it is and not switch teams. So just be aware of that. So these are kinds of growth. And I'll let you screenshot that if you want. You can just go ahead and pause and screenshot it. So we also have different kinds of cell death. So cells aren't supposed to be immortal, although there are ones that show some evidence of that. If you look up HeLa cells in the book The Immortal Life of Henrietta Lacks, you might find that. So death comes in two flavors. One is pathologic, which means, again, evidence of disease. And so we've already seen this. It's necrosis. So in this process, cells burst and they spill their lysosomal contents all over the place. And that creates tissue liquefaction, which is exactly as gross as it sounds like. It's also stinky due to the hydrogen sulfide and other smelly compounds that bacteria make, and just gross in general. And then there's normal, or what can be normal, which is apoptosis. So sometimes cells are supposed to die. So for example, you are covered in dead skin cells on your surface, which are keratinocytes that filled up with keratin and then died, and that's what gives you a nice hardy integumentary membrane that protects you from the outside and keeps you from losing water, yada yada. But those cells had to undergo apoptosis to die and become what they are today. The difference between apoptosis and necrosis is that these cells are programmed to die, and they die in a compartmented way. So they don't burst. Instead, they end up being little cell fragments. And the lysosomes in each of these are going to combine with the organelles and produce sort of a homogeneous slurry of cell parts. And then another bigger cell, like a macrophage, can come and eat this up and just dispose of it properly. And then these are dead cell bits, but they're safe because they're not spilling all over the extracellular fluid. So please note, it's not appropriate to conclude from this that cell growth is good and cell death is bad. We all want to control which kinds of cell growth and death are occurring and where they occur and when, and that's how healthy tissue maintenance, regeneration, and removal happens. So an example of apoptosis that does something really cool is during your fetal development, you started out with a hand that looked kind of like this, a nub or a paddle. And inside of that nub or paddle, you had the precursors of your hand bones. but you didn't have fingers yet. But then the cells in between your fingers, which I'm going to mark as being yellow, started to undergo apoptosis and basically etched out your fingers until you end up with a little fetal hand. With actual fingers instead of just being a nub paddle. So the reason you have fingers that are separate from each other is thanks to apoptosis. So some kinds of cell death are good. Okay, and that concludes the video for the second part of the Chapter 4 tissue system. And I will keep recording these as fast as I can and get them up to you because this is how we learn. So I hope you enjoyed this video and it was at least entertaining, and I will see you in the next one.